So we will now be opening the open forum. Uh, if I may do a screen share, I can flash the questions so far for our okay. So we have gathered questions from our audience. Perhaps we can start with questions for Dr. Balolong. Dr. Balolong, there is a question from Dr. Asuncion Raimundo. If there are studies on build-up of AMR genes due to continuous use of disinfectant in the industrial settings?
Hi, Ma'am Sean. Thank you for your question. Actually, there are some relevant data, not only in WWTP, but also in other environments such as soil, for instance, because antibiotics are not the only stressors in the environment that would contribute to resistance. So, for instance, disinfectants and other chemicals that we continuously use, those act as the stressors and microorganisms will pose countermeasures. That's why I mentioned a while ago that there are the intrinsic ones and then the acquired. So all of these stressors may trigger either the acquired resistance of microorganisms to resistance, either resistance to antimicrobial, resistance to chemicals, or resistance to other stressors, ma'am. So there are papers already that describe such build-up of genes. Build-up meaning the number of subtypes or categories of AR genes accumulates, and also, ma'am, not just accumulate, no, but sometimes they form like shifts, or there are some parang nag-iiba yung pattern whatever stressor would come or would be present in the particular environment. I hope I answered the question of Ma'am Sean.
Thank you, Dr. Balolong. There's also a question from Nicole regarding the most comprehensive database available that can be used uh, as reference for identifying resistomes.
Yes. Currently, actually, there are many databases. If you would Google, no, there are many databases, small ones, no. But starting this year, they have what they have. How can I state it? They have ano tawag dito, they have made or developed this card 2020, no. So this particular database that we can access at the NCBI actually will contain all those small databases. So parang yun na yung comprehensive AR database that we can use. And it runs in Ubuntu, and you can access through NCBI as well.
Okay, and another question is from Paul Christian Gloria.
Mhm.
He wants you to comment on probiotic, prebiotic, or symbiotic interventions for humans. Can this be the future of protecting us against AMR derived from the environment?
Well, as a scientist of probiotics, I wish, no, but of course, I cannot directly say right now that they can. Of course, we have to build evidences before we would say yes, no. But of course, we will be considering that particular idea. That would be interesting.
Thank you, Dr. Balolong.
Thank you.
Our next question, before we go to the questions for Dr. Papa, I think this question can be answered either by Dr. Balolong or Dr. Bandoy. So the question is, do you think antimicrobial resistance has something to do with the COVID-19 pandemic? Dr. Balolong or Dr. Bandoy?
No. uh, uh, completely different realm, na completely different realm. I think the AMR has a different story to tell, no? Para may sarili siyang libro than the pandemic, no? The viruses have also a different track to employ. For me, that's my opinion. Magkaiba sila. They have nothing to do with each other.
Okay. Thank you, Dr. Balolong. Dr. Banoy, do you want to comment on
So
the question?
antimicrobial resistance is in the bacterial realm. COVID-19 is due to a virus. So they are different in mechanism. So although may resistance din sa antivirals, but they are different and unrelated to one another.
Siguro, Dr. Les, what would be parang ang kanilang scenario na pareho is that we can get AR bacteria and AR genes as well as viruses from animals or from the wild animals, and then it can be transmitted to humans, you know, particularly the zoonotic viruses. And there are some AR microorganisms that have been found in wild animals. Siguro yun. Pero in their realm, magkaibang mundo sila ng tinatahak.
Yes, these are really two different things. Perhaps the question stems from the One Health context it stems from the One Health context. That's why one of our participants is asking about, is there a possible relationship? Okay, so perhaps we can now proceed to questions for Dr. Papa. For Dr. Papa, Dr. Raimundo is asking if bacteriophages can be applied to control biofilm formers in the pipelines in the food and pharmaceutical industry.
Yes po. Thank you, Ma'am Sean, for that question. Yes, actually po, bacteriophages are now being used, well, of course, in other countries, for biofilm formers in catheters po. No, this is for the medical industry, but for catheters, they have been published already. It's been going on. There is an ongoing study in Europe wherein they're also trying to use bacteriophages for biofilm formers in underground and pipelines po. But I only saw that in a presentation in a conference. I haven't seen any published paper, but of course they can be used to control biofilms in pipelines, in food, and also pharmaceutical. Pwede po sila.
Okay, Dr. Papa, how about the question of Dr. Ronilo Flores about the potential of reverting back to phage therapy for treatment of bacterial infections with the emergence of the AR.
Yes, thank you, Dr. Flores, for this question. Actually, that is maybe the dream for a phage biologist like me to revert back. Although that's why we try to do these kinds of studies, no? Parang feasibility studies itong mga ginagawa namin. So that also to prove that, you know, phages can really be used and are really effective. But of course, there are a lot of challenges that we will be facing. So hopefully we can revert back. But yeah, because of course there mas mura siyang i-produce yung bacteriophages, and but the challenges, one is that we lack phage biologists in the country. Konti lang po yung mga nagtatrabaho sa phages, and that there are also in the country there's no regulatory board for yet, no, for the use of phages. So just a little of the challenges that we will be facing. But yes, I see, you know, later on the potential of reverting back to the use of phages for therapy. I hope I answered the question.
Yes, Dr. Papa. Now, the third question is from Paul Christian Gloria. He's asking if you have conducted long-term studies regarding CRISPR immunity in the tested bacteria killed by the phages, and if you have seen anti-CRISPR or ACR genes.
Okay, thank you for the question, Sir Paul. We haven't, in my lab, no, sa lab namin sa UST, we haven't conducted any CRISPR studies pa. But I came from, for my internship in Quebec, I come from a CRISPR lab. No? My boss and my advisor, Dr. Sylvan Monu, is actually, well, it's a CRISPR lab. So, I have not done CRISPR studies, but we are in the talks of eventually, hopefully, working on this kinds of on CRISPR. I have a lot of lab mates in Quebec wherein they have worked on CRISPR, and if you can look for papers by Dr. Sylvan Monu, they have work on different genes, even ACR genes, and CRISPR. We, in the lab, ang ginagawa pa lang namin is that we are producing mutants. We call this the bacteriophage insensitive mutants, or BIMs. Usually kasi when the bacteria has the CRISPR-Cas or a resistance mechanism, usually CRISPR is seen. No? So what we do is that we produce the mutants, we test them with our bacteriophages, our library of bacteriophages, so that just in case magkaroon ng mutation, then we can readily address this resistance, no, by the wild type reverting, going into the mutant. But so far, to answer your question, wala pa po kaming ginagawa on CRISPR. But we're going there, hopefully. I hope I answered the question also.
That's exciting, Dr. Donna, to venture also into CRISPR. So let us have the next set of questions for Dr. Bandoy. Aside from this question, there are also questions in the chat. But Dr. Bandoy, you are asked about what do you think is the main driver of the mutations of the virus?
Several factors, but for RNA viruses, they don't have the correction mechanism, so it's error prone plus immunological selection. But it's primarily the error prone property of the RNA viruses is accounting for the majority of the mutations.
Thank you, Dr. Bandoy. And a question from the chat box is asking you if you can expound more on the implication of COVID-19 genomics on our situation in the Philippines. This is from Dr. Michael Prementilla.
One of the key factors that driving this pandemic is the super spreader. So we need to do more. So if we cannot do whole genome sequencing, what we can do is contact tracing, because once we were able to identify super spreaders in hospitals, in the community, we would be able to stop the pandemic. And what that tells us is that community lockdowns are not as effective as identifying. So it does not give us more protection by having community-wide lockdowns as much as we can do contact tracing and identifying the super spreader. That is the insight from the genomic study. Super spreaders are really driving the pandemic right now.
Okay, thank you for that insight, Dr. Bandoy. There's another question from Dr. Ronilo Flores regarding if you would consider the doubling time or the R value as a more robust basis for interventions or policies related to COVID-19 in the Philippine context.
So, from the UP Pandemic Response Team, we are advocating a use of multiple metrics: case fatality rate, reproductive number, absolute number. Because even if you have a low R, if you have 20,000 confirmed cases, active cases, that's something that has to calibrate the reproductive number. So we advocate multiple metrics so that we have a holistic point of view of the overall situation of the pandemic. So not just one metric, but for the doubling time, you need to calibrate it with the absolute number. So even if you have low values for that, but the absolute number is large, it still is a cause for alarm.
Okay. So Paul Christian Gloria also has a question for you. He's asking why nanopore sequencing is not being used in the Philippines. Would you know why? Because is this not a suitable technology as a virus has a relatively small genome, and nanopore technology is cheaper and smaller?
In our laboratory, we are focused on sequencing, and nanopore is one of the technologies we are exploring. It's used by RITM. So the sequences submitted by RITM is nanopore, but it's not fully adopted because of it's a relatively new technology, and there's actually a bottleneck in the trained um, molecular biologists employed in the testing lab. That is a gap. So we have a gap in manpower. Most of the people who are doing the testing has just been recently been trained. That's the information that I get. So nanopore, so that's just PCR, and nano sequencing is much more complicated than the PCR that we are doing. So there are gaps in the technology and gaps in the manpower.
Okay. Perhaps we can go back. Thank you, Dr. Bandoy. Perhaps we can go back to Dr. Papa. There are additional questions for Dr. Papa. Kevin Dagbay would like you to comment on the general safety profile of phage therapy and the origin of molecular specificity of phages to a specific strain of bacteria.
Okay, so thank you for the question, Sir Kevin. Well, for the general safety profile of bacteriophage therapy, since phage therapy has been used already for decades, no, since 1921, 1920s, Principi et al., there's this paper by Principi et al. In 2019, wherein they gathered all the publications that have been done for bacteriophages for therapy. So there are more than 100 papers that was analyze and check. And so far, with all the publications that have been done, like I mentioned a while ago, bacteriophages are known to be safe to eukaryotic cells since they are very specific to bacterial cells only. They do not jump to a eukaryotic cell, like a plant cell, or even to an algae, or even to a nematode. So with that, and also when the phages are administered into the body, usually bacteriophages do the job immediately. Okay, so when they have in contact with the bacteria that is causing the infection, usually within 30 minutes, they can already do the job in eradicating the bacterial organism. So far, there are no reports on the immune response for the activity of the bacteriophages. Maybe because one, they do the job immediately and it's done. No? So far, with all the studies being done and reported and published, bacteriophages are safe. But of course, there's that concern wherein just in case your host organism will mutate, then bacteriophage should also be able to address that problem. Now, for the second question, molecular specificity of bacteriophages. Well, there are receptors that are usually found that are compatible between the bacteriophages and the bacteria that they infect. No, for example, there are specific receptors for salmonella, protein receptors, but sometimes salmonella typhi and other salmonella, it might not be the same. Okay, so that's why that is one of the major importance of bacteriophages, because they are very specific, sometimes even to a specific strain. It may be a disadvantage also, but if you want to just direct your treatment or your study to just one specific organism, then you can look at the protein receptors. You can look at the specificity of these bacteriophages to their certain host. So I hope I answered the questions.
Thank you, Dr. Papa. So it's already 10:30, so we are wrapping up our question and answer session. So I would like to thank our speakers for this session. This has been a very enriching and very interesting session this morning. So thank you to our resource persons, to our speakers, and to our participants for attending this session of the 2020 APAMS. I would like to turn over to uh, my co-host in this session, Dr. Michelangelo Promentilla, to formally close the session.
Thank you, Professor Dalmasio. I'm Les for moderating the second half. And again, I would like to thank everyone for making this webinar possible. So words are not enough to express our gratitude to our resource speakers, from our speaker from NTU, and of course speakers from the Philippines. In fact, if I had to sum up this session, I would say it's quite diverse. It's diversity, and this diversity will hopefully translate into potential collaboration so that we will not work in silos. I mean, I think material scientists, engineers, biotechnologists, microbiologists would work together for a holistic vision of for example, of One Health, Eco Health, and Planetary Health. And I think the topics here, if you would like to connect the dots, is actually toward to this vision. Again, I would like to thank PAASE for allowing us to host this webinar. Ma'am, Professor Giselle Concepcion, the PAASE president, of course, Dr. Kathleen Aviso, who's helping us to manage this webinar. So thanks again to the PAASE chair and co-chair for having this kind of webinar. So I think it's I had to close the session, but before closing the session, may I ask Dr. Kathleen Aviso, the co-chair of the this 20th APAMS, to provide some advertisement.
Yes, okay. Thank you very much for that, Mike. So allow me to share my screen. So essentially, I just want to inform everyone about the other sessions of PAASE. So for those of you who are not familiar with the organization, so this is the Philippine-American Academy of Science and Engineering, and we are actually celebrating the 40th PAASE anniversary together with the 2020 annual PAASE meeting and symposium, of which this particular session is part of. So we started our session since July 20, and we're running all the way to August 14. Every day we have sessions and lectures from Mondays to Fridays from 8 o'clock in the morning, extending up to 10:30 or 11 in the morning. So if you're interested, so kindly scan the QR code over there, here at the bottom of the slide. So it will take you to the program of the entire program of PAASE for the coming days. So I'd like to plug our sessions for tomorrow. So we have 3 different parallel sessions. So you may want to attend or participate in some of them. So we have diversity and representation in STEM and higher education. So that's parallel session 7.1. So you can see here how you can register together with the link to the YouTube live for that particular session. We also have the second parallel session, which is on electronics, electrical engineering, and instrumentation. And then finally, we have the third parallel session on physical and geological sciences, also tomorrow at 8 o'clock in the morning. So I hope those things would be of interest to you. And again, you can visit the program so that you can view other upcoming sessions. So we are present in Facebook. So you may look at our webpage, which is at Philippine-American Academy of Science and Engineering, Facebook official PAASE. And we also have our official webpage, which is at paase.org. And we are also conducting webinars on a regular basis on Fridays. We will resume after the 2020 APAM Symposium is completed. So all of our webinars are recorded and uploaded in YouTube at PAASE Webinars. So that's all from me. Okay. All right, Mike. I'm not sure if you have anything else to say.
I think, Kay, do we
Yes.
have a photo shoot for this?
Oh yeah, sorry about that. Okay, can I have do you want everyone, including the participants, or
we have
can
to
we have
turn
the
on all the cameras, right?
okay, can everybody please turn on your camera so I can do a screenshot? Okay, let me see, have a look. All right, so we have a total of 53 participants, so I'll be doing it several times, so kindly hold. Okay, so I'll take the first batch. Okay, hold on, sorry. Okay, I'll take the first batch. And 1, 2, 3, smile. Hold on as I toggle through the next section. Okay, for the next batch. Okay, so everyone smile. Oh, can I have everyone to please turn on their videos? Yeah, not just the speakers. Can I have the participants turn on their videos? Yeah, so that I can get a screenshot, not just the names. Okay, thank you guys. All right, I have a few more who haven't turned on their videos. So I'll give you 10 more seconds. 5, 4, 3, 2, 1. Okay, I'll take a screenshot. So 1, 2, 3, smile. Okay, thank you. Yeah, I think that's it. I think we have all the okay, I have the shots. Okay, although some of you did not open your videos. Okay, that's it, Mike.
Okay, thank you, Kate. Bye, everyone. Have a delicious lunch.
Hi,
Thank you,
everyone.
guys. I hope you can participate in our other sessions.
Bye.
Thank you, PAASE, for inviting. Sir
Bye.
Mike, thank
Thank
you.
you.
Bye. See you around.
Thank you, Mike.